Stories. All our lives, we're surrounded by them. They come in the form of myth and religion, as books or films, or even simply as the personal stories that we tell about ourselves. Stories have always also been used as a form of knowledge sharing. You may think that we have left the age of storytelling when we entered the era of science, but I don't think so. I believe that every scientific discovery has a story to tell us, a true story about the nature of our world. We're here today at the University of Zurich to give a group of young researchers the chance to tell us their most exciting science stories at the FameLab semi-finals. FameLab is an international science communication competition that started in 2005 in the UK and has since spread to 31 countries all around the world. The rules are simple. The contestants have 3 minutes to explain a scientific topic of their choice in a manner that is understandable to everyone. They are not allowed to use any slides or electronic support and only use the props that they can bring onto the stage with themselves. They will be judged on the 3 C's: content, clarity, and charisma. The 3 winners of today will move on to compete in the national finals in Bern in September. Let me present the jury for today. We have Dr. Sarah Carmignani, a biochemist, counselor, and part-time science writer. We have Professor Helmut Gerhardt, a stem cell and cancer researcher at ETH, and Professor Daniel Kieper, a neuroinformatics professor at the University of Zurich and director of the Life Science Zurich Learning Center. But, of course, the most important people today are our contestants. So let us dive right in and hear their stories about everything from neuroscience to silicones, and let them take us on a journey through the beautiful world of science.
Fatigue. Take a moment to think about what that word means to you. You might be thinking of being beat after a sleepless night, drained after a hard day at work, or physically worn out because of an infection. And right there, you realize that fatigue can mean many different things for different people. Fatigue goes beyond just being tired. Fatigue is a form of extreme tiredness that is not relieved by typical recovery strategies such as sleep. It is present in a wide variety of disorders, including long COVID, and is a creature that has multiple faces. You might experience the more physical effects of fatigue, the more mental effects, or be unfortunate enough to actually have both. It is believed that fatigue does not arise from one single cause or mechanism, but that it is actually the product of several pathways that can coexist. Our research group is particularly interested in one hypothesis involving something called metacognition of control. Now, I know that's a bit of a mouthful, so let me explain what I mean. Metacognition of control is the brain's system to self-monitor the control that it has over the body. And indeed, body and brain form a closed loop, whereby the body sends information to the brain, that information is processed in the brain, and then the brain sends signals back to the body to exert change. Now, imagine that you are outside in the winter on a chilly Swiss mountain. Your body is going to send signals to your brain telling it that your core temperature is sinking. Your brain is going to process that information and send signals back to the body telling it to start to shiver in order to produce heat, but also to take on action, such as taking out that jacket. In disease, one or several parts of this loop might be affected, and this can generate either wrong information, wrong information processing, or defective signals. The brain's self-monitoring system would see this. It would see that something is constantly wrong, even though the brain is trying to do its job well. And our hypothesis states that the brain might then conclude that all actions are pointless, and that it might fall into a state of learned helplessness that is expressed as fatigue. Now, like I said, this is still a hypothesis and something that we are currently testing in a study that we are conducting with patients with multiple sclerosis. Our hope is to better understand the mechanisms behind fatigue in order to offer better and more targeted therapy. Thank you for your attention.
Sounds, they're everywhere. And we perceive them here. For most people, it makes sense that processes like decision-making or logical thinking, memory, happen in the brain because we need to think for them. But for our senses, the thing that the brain is involved is not that self-explanatory. I mean, we have noses, eyes, ears, tongues. We have actual organs for all of our senses. And we perceive those senses in exactly those locations. We taste here, we smell here, we see here, and we hear here. Or do we? Well, I mean, this very unsuspicious question already lets you guess. We don't. Our senses are not confined to their respective organs. The brain is involved. Glass is clinking. Now what happened here? Sound waves. Sounds are transmitted as waves, and they contain informations. Volume, duration, intensity. Our ears capture those sound waves, and through a very elaborate mechanism of amplifying and transmitting, those mechanical wave information are converted into electrical signals that our brain understands. Now, our nervous system is built up of cells called neurons, and those neurons have emerging long cables called axons, and through these cables, these electrical signals are transmitted and the neurons can connect to each other. Now, our journey of this auditory processing starts in the ear with a very long cable that goes into the brain. Our first stop is the brainstem. That's an area that connects the spinal cord to our brain. And there some basic processing happens. Volume, pitch, and duration. A bit higher up in the brainstem, the location of the sound is determined. And the higher we go, the more complex features of those sounds are determined. Eventually, we end up in the primary auditory cortex. That's the outer folded layer of our brain. And there, our sound steps into consciousness. By connecting with other brain areas, our sound is getting interpreted and associated with previous knowledge. For example, the human voice is processed right here in the temporal lobe, where also this primary auditory cortex lies, and we understand the words a bit far back in the Wernicke area. So, the actual hearing that we consciously perceive is not happening in the ear, but in the brain. However, without the ear and its inner parts, there wouldn't be any signal for the brain to process. So remember, hearing goes beyond the ears. And now, all that's left to say is, thank you for hearing me out, and cheers.
So, hello everyone. So, let's talk about some very interesting materials that we use in our daily life. These materials are called silicones, or technically in the science we call them polysiloxanes too. Let me start with some examples we use in everyday life. The cooking ware. Silicone, we cook very nice curries with it. Second example, we bake very nice cakes. It's also very flexible using silicone molds. Third example is, I will not tell you what it is, but it makes people happy. It's for pleasure applications, also silicones. And the fourth example is lubricant. It's also for pleasure applications. It makes people happy. So let's get back from happiness to the science of silicones, like what makes this thing so thermally stable that we can bake the cake or we can use them as lubricants. So it all comes here. If you see, this is a chemical structure of the material. It's a semi-organic material, so it has organic carbon, if we see here, and it has silicon and oxygen atom. Silicon and oxygen atom makes the material flexible, and its chemical nature also gives it softness. That's why the material is flexible and soft at the same time. Now, how the structure looks like for this material? Actually, it's like spaghetti. Like we eat the noodles, or let's say we pasta, it's like spaghettis. This material is normally liquid at room temperature. That's why the lubricants are liquid. And it has a random structure like I show here. But when we can chemically react it, and when we chemically react this material, so all this spaghetti structure can react with the very special reaction, and then it becomes a cross-linked structure. So it's like, let me show you, it's like a cross-link. So this is a silicon chain, like spaghetti, this is another chain, and it's with this chemical reaction, it just cross-links with another molecule. So this makes the material solid and makes it thermally stable. So that's why we can use it for baking or cooking curries and many other applications in daily life. So I hope you like this short introduction on the silicones. And now try to find what kind of silicones products you are using in your daily life and have fun with the silicones. Thank you very much.
Have you ever heard of the saying, comparing apples to oranges? It means that comparing two fundamentally different things is practically incomparable. But whenever we go grocery shopping in Migros, we often compare apples to oranges. Do I compare this apple over an orange? I study brain activity during simple choices using a powerful tool called functional magnetic resonance imaging, or fMRI. fMRI measures indirectly neural activity through the blood flow in the brain. More blood flow means more energy in that brain area is being used, and more energy means more neural activity. We find that an area in the brain called the prefrontal cortex lights up whenever I'm deciding between an apple and an orange. And in making that decision, the prefrontal cortex basically collects subjective information, and which we call as subjective preferences, and this is drawn from our experiences. I personally prefer an apple over an orange because, to me, apples are tastier than oranges. But preferences are widely different to different people, and fMRI can measure these individual differences in brain activity. A limitation with fMRI data is that it's correlational. The prefrontal cortex could well be involved, but so do other brain regions such as vision, memory, and attention. If we can disrupt prefrontal activity and observe impaired behavior, then we can have causal evidence that the prefrontal cortex is indeed important during decision making. In our lab, that's exactly what we did. Ethics won't allow us to cut out a person's prefrontal cortex, but we were able to momentarily disrupt prefrontal activity using brain desynchronization using an electrode over the prefrontal cortex. Remarkably, we find that desynchronizing prefrontal cortex led to less consistent choices. In other words, I prefer an apple over an orange now, but with a desynchronized prefrontal cortex, I end up randomly switching my preferences between apples and oranges and so forth. The prefrontal cortex is important during simple choices. It's also important whether I want to buy a house in the suburbs or an apartment in the city, or choosing which leaders that will best represent our future. These are difficult real-world decisions, so difficult it's like comparing apples to oranges, or more like the prefrontal cortex is comparing apples to oranges.
From embryo to adult and back again. Do you know how many genes and signaling pathways and interconnected networks we need to come from a single cell to an individual? I don't. I just know that there are too many, and it's a complicated journey from a single cell to an adult organism. These pathways and genes that are active in embryo, they are normally silent in the adult because they're no longer needed. But now we know that in some diseases like cancer, some of these programs are reactivated. Have you heard of metastasis? It is when tumor cells leave their primary site, go to the circulation and go to other tissues, and they make secondary tumors. That's when it's already too late. And unfortunately, we don't have effective therapies for metastatic cancer, for most of metastatic cancer. And the reason is that there are many genetic programs that are activated in these cancer cells that metastasize that are not present in healthy tissue, and it's very difficult to understand them. And guess what? Many of these genes and programs are the ones that are active during embryonic development. It's like embryo hijack, cancer hijack these genetic programs from embryo and make an independent individual inside patient's body. And it is very difficult to understand unless we go back to embryo and we study those pathways in their original context, which is embryonic development. That's my research. That's what I do. I use embryonic development side by side with cancer research to find these common pathways, to understand them better in embryo, and hopefully to be able to target them for a better treatment. And that's from embryo to adult and back. Thank you.
About 1 billion years ago, life was rather boring in the ocean for the bacteria. But something unexpected and a beautiful event happened that changed the course of evolution forever. Somewhere in the ocean, on a bright, sunny day, this big bacteria meets a small bacteria, and it decides to eat it. But surprisingly, the smaller bacteria survives, which results in this long-lasting, mutually beneficial, romantic relationship that lasted thousands of years. And through the course of evolution, this smaller bacteria evolved into what we now know as mitochondria. And the bigger bacteria evolved into millions of species on Earth, including me and you. Hello, and welcome everyone. On the other side, I'm Sandeepan, a PhD student in the Basler group at University of Zurich. I work on Drosophila development, and I'm very excited about mitochondria and science communication, and I'm here to convince you of the importance of this amazing microscopic structure in our daily life. Mitochondria is very important in aging, in disease conditions, in lifestyle and health. Diet is very important, and what we eat decides how our mitochondria is. Apart from the role in energy production, scientists have now uncovered the role of mitochondria in development, in embryonic development, in adult development, and also it plays a central role in aging and also in cancer. What we eat decides how we age. What we eat decides how the disease conditions progress. And our lifestyle choices, like exercises, also influences mitochondria. Mitochondria is known to exist in two different structures. One is where they are these large fused tubular structures, and the other where they are smaller fragmented structures. The balance between these two structures is extremely critical, and this balance is maintained by the diet and by the lifestyle. So if we follow a good diet and a good lifestyle, we will be able to maintain good mitochondrial quality. Research has shown that sleeping behavior is very important for reducing these so-called reactive oxygen species from our brain and body, and their mitochondrial health is kept good. Therefore, what I am suggesting here is mitochondria plays a huge role. Mitochondria plays a huge role in our daily lives, and if you eat healthy, if you do exercises, then you will have a healthier mitochondria and a healthier relationship, just like our bacterial ancestors. Thank you.
What a great selection of talks, and what a wonderful group of storytellers. I hope you have been as entertained and impressed by them as I was, and that maybe you've learned a little something along the way. Before I tell you which 3 contestants will be moving on to the finals, I want to thank a few important people. First of all, let me thank all the judges who took the time to listen to all of the talks and give their impressions on them. Then I want to thank Life Science Zurich for organizing and supporting the FameLab competition here in Zurich. And finally, my warmest thank you goes out to Silvi Kuparos, who is the main organizer for this event, not only in this year, but also in previous years. As a previous contestant myself, I know how much opportunities like this one can mean to people. And I'm very glad to see that this year another group of young researchers got the chance to participate in FameLab. So let us finally find out who won the day. I'm sure that every one of you has their personal favorite among the contestants. And I'm also sure that with their different styles of presentation, every one of them has made a little fan club among the audience. But in the end, it is the jury's vote that counts. And they have decided that the winners of this year's Zurich semi-finals of FameLab are Miguel Garcia, who impressed the jury with a great stage presence and a clear explanation of the complex neuroscience of decision-making, Florence Steiner, whose awesome auditory introduction made us all ears for her talk on hearing, and Kushdeep Sharma, whose contagious enthusiasm for silicone shone even through the screen. Congratulations to all 3 finalists, and good luck at the national competition in Bern.